After translation, polypeptides are processed. This might include cleavage, breaking them, cutting them apart, maybe even chemical modifications. The length of functional time of a protein is going to be regulated by selective degradation. Cells mark the proteins that need to be degraded because, of course, they're going to constantly be recycling the materials that they're made of by attaching the substance ubiquitin. This mark, this ubiquitin mark, is recognized by what we call proteasomes, which recognize and degrade appropriate proteins. There's a small amount of DNA that codes for ribosomal RNA and the transfer RNAs. We used to call the DNA not coding for protein junk DNA. Not anymore. We think that it probably has significant importance, and it is a significant fraction of genome, especially for these production of non-coding RNAs. We still are continuing to research what is this DNA and why are so many parts of it repeated, and what is the functioning of the introns and the RNAs. So let's talk about some of those non-coding RNAs. The first ones are the microRNAs, and they are small and they're single-stranded, and they bind complementary sequences in messenger RNA, and they can actually cause the degradation of the messenger RNA, or they can block translation of it. And we think maybe half of all human genes may be regulated by these microRNAs. It's very interesting, very precise control. We also have what are called small interfering RNAs, and they're similar to the micros in size and in function, and they can block gene expression. And this is called RNA interference. And this is really interesting because in the lab, you can actually use these small interfering RNAs to turn off a gene function and then maybe discover its actual function by doing so. Some non-coding RNAs can change the structure of chromatin. Boy, we continue to talk about that, don't we? It's very, very important. In some yeast, for example, small interfering RNAs can reform heterochromatin at the centromeres after chromosome replication. Pee-wee interacting RNAs induce the formation of heterochromatin, and that completely blocks the expression of parasitic DNA known as transposons. This also helps to reestablish the correct methylation patterns during gamete formation in many animals. Long non-coding RNAs, they have hundreds of thousands of nucleotides. One of them is responsible for the X chromosome inactivation, and we think that it may also help regulate chromatin structure and play a role in gene regulation. Small non-coding RNAs regulate gene expression in many ways. If you increase microRNAs in species, then it allows you to increase morphological complexity. As far as their evolution, we think that the small interfering RNAs came first, followed by the microRNAs, followed by the pee-wee RNAs.